So as you know, we have this in an open format. And so from the perspective of please try to keep your own computers muted. We wanted to be able to allow everyone to have interaction with our speakers later on. So this format's a little bit better than the webinar features in Zoom. So thank you for tuning in. Why genetic matters, the confirmation of the number of different genes that go along with NM really allow us to plot the future course for therapeutics. So we'll go to the next slide. Simple housekeeping, closed captioning is available if you're interested. This webinar is being recorded and will be shared later on. Please keep yourself muted until we get to the discussion section, and then we'll end with Q&A. All right, so just for the group here, we're going to try and take a quick poll. Obviously, this won't work on the recorded set, but if you actually are here and have nephilomyopathy, or you have a loved one with nephilomyopathy, we'd like to know if you have a confirmed genetic diagnosis. So not only a diagnosis that came from a muscle biopsy, but one that actually has a genetic code listed for it. So if you could take a moment to kind of click yes or no, that'd be great. We'll have the results here in just a second. And Kate's manning the ship here, so thank you to Kate for administrating over the Zoom.
I'm going to end the poll, so if you have an answer, to go ahead and click yes or no, or I don't know now.
Don't forget to press submit. That's great. That tells us quite a bit. I think, Kate, if you have the numbers of folks that responded, that'd be great. I'm not sure if you can see that on your end. And we can move to the next slide too. So the entire grant from PCORI has been founded on the idea that when patient communities get involved in research, that the research has moved along more progressively. And so that is the funding is completely about trying to get the community involved, to actually talk to the researchers, to actually influence how research will be advanced. And the steps we take are meant to be inclusive of all those with NM, not just the ones that we find in sort of the middle ranges, but even the ones that are the more rare forms and at any age. So please be aware that we're trying to be inclusive to all those with NM. So the quick agenda, I'm going to do some reminders of some strategic goals that the foundation has, and then we're going to have both Brianna and Karsten speak about the genetic confirmation, the subtypes, as well as the therapeutic toolbox, and then we'll wrap with the Q&A. We'll hopefully be trying to be done within an hour. So we had an original research timeline that we produced, I'd say earlier this year, and I'm going to take you through an amended one on the next slide. We'll share this deck with you, but here's an updated research timeline. So, this year, we're completing a major CRISPR project, both in the actin variety and NEB varieties, that are giving us a lot of information about how well that therapeutic target can work. That work's being done both by Rhonda Taylor as well as by Jim Dowling. We're going to understand what medicines cause NEB zebrafish muscles to be more active. That's the research we've done down in Australia as well. We already hired and trained a representative, Sam Jacobson, for the foundation to actually represent us with the CMDIR as a coordinator to add and update CMDIR registrants. I think we've talked about why having a robust registry is important. The industry, biopharma, is going to want to know that we are prepared to move and launch into clinical trials by being able to contact our community. So having those registered and genetically confirmed will allow us to kind of build a case with these pharma companies to be able to therapeutically target each specific problem. By next year, we hope to have 2 industry-based companies, well-funded companies, actually testing their individual innovation and therapeutics on NM tissues. And we've got a nice start on that already. We have, we're going to start data collection, I hope. We have a meeting about this on November 30th, but we're going to start data collections and clinical exams for our US-based natural history study in 2023. It's likely that we'll ask you, the community, to step up and participate in some of those exams, if not all of them. And that'll be probably a 3-year course that may require upwards of clinical intervention, either virtual, but mostly in person, about every 6 months. We hope to increase our number of registrants that are confirmed genetically diagnosed to 400 people. Right now, we're at 265, but a fair bit of those are not confirmed. So we'd like to be able to hit 400 confirmed registrants by the end of 2023. And that'll be with the help of Sam Jacobson as well. In 2024, we'll be completing our first year of the natural history study. We'll test 2 interventions on more rare forms. One of those projects is already underway. We hope to reach 100 tissue samples in the CMDTR, the tissue repository, where our community is able to contribute some tissue collections so that when researchers need human tissues in the Petri dish to be able to test different therapeutics, that will be available to them. By 2025, we hope to complete the final year of the natural history study, identify optimal treatment therapeutics for clinical trials, and establish interaction with the FDA to agree on the protocols and pathways for regulatory approval. In 2026, we want to launch with industry, an industry partner, a supportive treatment, and then finalize the discovery of genetic repair and a curative mechanism. So that's again, a 4 to 5 year timeline that we'd like to try and have you, the community, hold us to. Jump the next slide, Kate, please.
One sec, I'm experiencing some technical difficulties. I think there we go, right?
Yeah, perfect. So one of the interesting learnings this year at SciFam was that, for me anyway, was that in 2020, just about 2 years ago, a new diagnosis code for Nemelin myopathy was established. And so many of us have never actually informed our doctors, nor have our doctors been informed that this code exists. So this is an ICD-10 code. So it's a diagnostic code that your physician can put in your medical chart. And that allows us to more specifically track where the NM folks are on the back end. There are vehicles where you can actually look at insurance claims data, and those insurance claims can tell us exactly where the NMers are seeking care and the type of care they're seeking. I see Joanna's quick message. I think it's primarily United States driven, but I'd rely on maybe Gus or Karsten to let us know if ICD-10s are exclusive to the US and to US claims. Gus or Casey or Brianna, do you know the answer to that one?
I don't know the answer to that one. I imagine that is the US only.
It does stand for international code, but I don't know if they're used in the same way.
Just so this is Sarah. Is that ICD codes are generally adopted by the World Health Organization and I think are used globally, but we can clarify that point. It's a great question.
Great. So what I would ask of the community is when you go to the doctor next time, you can ask if this code G71.21 actually is registered within your medical chart. We've used a system recently called Komodo Health that has identified 500 people with Nemelin myopathy in the United States already utilizing this code. And we can actually see which medical centers they're actually attending. And that would allow us to reach out to those practitioners to get more people involved in a clinical trial eventually. So if we actually reached out to all those doctors saying, we know you have this many NMLA myopathy patients, we've seen you use the code, would you be able to reach out to them directly to get them engaged in our clinical trial? So anyway, just so you know, that is a code that we're looking to actually make more robust over these next couple of years. So anytime you do see your doctor, please try to utilize this code in your medical chart, G71.21. All right, so the speakers today, the least intelligent guy on the top, the most intelligent follow, Karsten Bonnerman. Again, so grateful that he's here. Senior investigator in neuromuscular neurogenetic disorders of the childhood section of the NINDS, the National Institutes of Health. Brianna Gross, neuromuscular genetics counselor at CHOP Hospital in Philadelphia. Gus, the scientific director and project lead for us here. Alan Beggs, you guys have heard speak before. Boston Children's Hospital scientific partner and genetics expert, and Casey Genetti, genetic counselor at Boston Children's.
So Brianna's speaking first, right?
Yeah.
Okay, give me 1 second so I can shift over to her slide deck.
Brianna, thank you again for being here. Really appreciate it. As we kick off, let's try to hold questions until toward the end. If you have anything, we can post them in chat and we can build a Q&A for later.
All right. Welcome, everyone. Thank you so much for coming today. It is a pleasure to chat with you all about genetics and genetic testing. So today's talk is called A Roadmap to Confirming Genetic Subtype. So we'll be taking some time to review basic genetics. If you could advance, please. We'll talk about basic genetics. I know that genetics can be a bit overwhelming, especially as many people have had their last genetics talk in high school or middle school. So we'll review basic genetics, genetic mutations, inheritance patterns. We'll also talk about genetic variations in genetic testing and their results that can come back from genetic testing. We'll talk a little bit about NMLA myopathy, and we'll also review a few other genetic phenomena that can occur. So genetics 101. In all of our cells, we have millions and millions of cells. We have a nucleus, which is the control center that tells the cell what to do and when. And inside of that is where all of our genetic information is stored. And our genetic information is stored in these structures called chromosomes. You can look at chromosomes under a microscope. They look like little X's, and they are we have 46 pairs, and they come, or I apologize, we have 46 chromosomes that come into 23 pairs. And they come into pairs because we receive one chromosome copy from our mom and one chromosome copy from our dad. The chromosomes house our genes, and we have over 20,000 genes that help us make us who we are. Our genes provide instructions for this. And if you think about a gene like a little instruction manual, there has to be some sort of language. And our genes use DNA or base pairs to provide these instructions and to act as the language. So instead of having 26 letters in the alphabet, DNA only has 4, which is A, T, C, and G. So what exactly are genes? We use our analogy that there are little instruction manuals. I apologize that my graphic got a little messed up here, but ultimately these genes provide the body with instructions to create a protein. And proteins are really what makes us who we are, from things that you can't see, like where cells need to migrate to things that you can see, like your hair color or eye color. So to review, we'll talk a little bit about a genetic mutation. The more specific way, and probably the more correct way to refer to a genetic mutation, is a pathogenic variant. Pathogenic variants or mutations, we will use these interchangeably, is a change in the spelling of a gene that causes the body to not be able to produce a protein properly. So here is a little example. So in my secret cake recipe, I have flour, I have baking powder, baking soda, I have the whole 9 yards, sugar, eggs, vanilla. But if I were to remove a couple of my ingredients, if I were to take away the flour, the baking powder, and the baking soda, I'm going to, you're going to try your best. You're going to get your mixing bowl, mix it all together. But ultimately, what you're not going to create is a cake. You're probably going to have something a little ooey gooey. Maybe it'll taste really yummy, but it's not exactly cake. And this is similar to what happens when we have a genetic mutation. Our body is going to do its best to use the instructions it has, but the protein product might not be exactly what the body needs it to be. So a little bit more advanced. So if we have a spelling error in our normal functioning gene, we typically the sequence is AATTCCGG. If we were to change that second T to an A, our body might not be able to produce the protein effectively. You'll see here that there's a little extra step, which is the RNA. You can think of RNA like the cliff noted version. So, when you read your recipe, you have to read it fully just in case that you don't miss anything wonky, like, for example, that you're supposed to mix the sugar with the baking soda or powder before you add your flour in, some odd thing. So the body has a little cliff noted version that cuts out all of the extraneous information and is a clean version so that it can ultimately create the protein. So as you can see here, if the DNA is abnormal, the cliff noted RNA is abnormal, and therefore protein isn't created effectively. So there are a few different types of basic genetic mutations or pathogenic variants that we can see. We could see a deletion, which is where we have the removal of a single letter of DNA or multiple letters of DNA. So in this example, the dog ran outside was changed to the ran outside. Duplications are where a single letter or multiple letters are duplicated in the gene. So we have the dog ran outside is switched to the dog ran outside. And substitutions are switches of letters. For example, the dog ran outside to the log ran outside. And all of our genes are inherited in a different way, meaning that in order for a person to present with symptoms, there can be a variation with how the genes look. So in autosomal dominant inheritance, only one of the 2 copies of a gene that we have will have a mutation within it, and that would be enough to contribute to symptoms in a person. The recurrence risk for this, or the chance that another offspring would have this change, would be 1 in 2 or 50%, because if you have 2 copies of a gene and one has a misspelling in it, you either pass along the one that has the spelling error or you pass along the one that's spelled properly. In autosomal recessive inheritance patterns, you'll see that both copies of the gene need to have a spelling error within it. Oftentimes, we'll find that parents are carriers, not always, but carriers are individuals who possess only one change out of the 2 copies that they have. And they themselves remain asymptomatic. They have another gene copy that is functioning effectively, and that prevents them from having symptoms. But when 2 carriers come together, there would be a 1 in 4 chance of passing along both copies that are misspelled and therefore contributing to symptoms in an offspring. That can also be looked at as 25%. Now, genetic testing. So genetic testing, there are many different types of tests that are available. And I will preface that not every genetic test is created equally. They all have different intents. And even within the same category of genetic tests, they might look a little bit different. So karyotypes is a genetic test that looks at a picture of the chromosome. So you can physically look at all of the chromosome structures to see if there's any full extra or full missing chromosomes, as well as big pieces that are missing or extra. You can even see, sometimes you see something called a translocation where pieces of chromosomes swap. It is way above my head to be able to figure that out, but we do have lab specialists who can tell if tops of chromosomes and bottoms of chromosomes are switched. To me, it all looks very similar. A microarray is another test that looks at chromosome structure, but this test can look for much smaller scale deletions or duplications of chromosome material. If you're missing a larger, not large enough to be picked up on a karyotype to the naked eye, a piece of chromosome that can include maybe one gene, a part of one gene, or maybe it includes multiple genes. We can have a single gene analysis where only one gene is looked at fully, and it is looking at the sequences. So it's looking at the DNA, the base pairs, to see if there's any errors within that one gene. This test is great when there is a known family variant that you are looking for specifically, or maybe a person is exhibiting a very particular characteristic that is almost pathognomonic with a particular gene. Panel testing is often utilized in congenital myopathies, which is where we select multiple genes that are all a little bit different, but have overlapping features so that we can cast our net a bit broader in hopes that we'll find a spelling error or so that we have the explanation for a person or family in that test. Not in this one in particular, not every test is created equally, and it's very important for your doctor to know what they're suspicious of so that you can make sure it is definitely included on the panel. Whole exome sequencing is another test that is commonly used in congenital myopathies, and this is where we're looking at the protein encoding regions of all approximately 20,000 genes in our body, which accounts for about 1.5% of our genome. This sounds very minuscule, but this test is often a very great test to utilize. The genes themselves have extraneous information, so not all of this extraneous information goes to creating that protein. So that's why it doesn't include, 100% of our genome. Whole genome sequencing is a test that actually looks at all of the extraneous information as well as the protein encoding regions of our genes. And this test is a little bit more challenging to order clinically, just because insurance companies often do not recognize this as a test that they would pay for. Unfortunately, it's a little bit more new compared to whole exome sequencing in the sense of people trying to order it now. I hope in the next few years, this will be much easier to order clinically speaking, but it is very much so available on a research basis. RNA sequencing is another way to look at genetic information. So if you remember, the RNA is the quote unquote cliff noted version of our DNA. So the RNA sequencing will be the tidied up readout version from our genes. And this is typically done on muscle tissue for neuromuscular disorders, not all, but most of them will be done on muscle tissue. So a muscle biopsy sample is needed, and they'll be able to see if the cliff note version looks different than what they would expect it to. So what exactly is reported out on a genetic test? The simple answer is variance. So a variant is a difference in the spelling of our gene, and we are made up of hundreds of thousands of variants from one another, and that's what makes us different. That's what makes us human. So an example of this, we can have a black 1968 Corvette or a red 1968 Corvette. We can have a 1971 Corvette and a 1983 Corvette. The they all can get you from point A to point B, but they all look a little bit different. Maybe they have different engines. Maybe one can go a little bit faster than the other. And we can classify variants or spellings, specific spellings of our genes by the way that they impact our body. In the laboratory, we'll report out variants so that we can determine if we have an explanation for a person's symptoms or not. Variants are classified on a scale. So on the left end, we have benign and likely benign variants. So this is something that you see very frequently in the general population. It's just variation amongst us humans. And this is not typically reported out on a genetic test because there would be hundreds of variants to look through. If there was ever curiosity, you can always request to look at all variations that were identified. That makes you uniquely you, but that is a very big list. The next variant classification is probably the most tricky, and that's a variant of uncertain significance. That's when the lab does not have enough information to say, yes, this definitely contributes to symptoms in a person, or no, this is just normal human variation. Ultimately, there's a lot of things that we look at as clinicians to help us determine our level of concern about a variant of uncertain significance. Sometimes we can easily say, this is definitely the explanation. Or sometimes we can say, looking at it, no, this is human variation. We don't have to think about it again. Sometimes we have to do a little bit more research. Ultimately, what we look at is, has it been seen in the general population? Has it been reported in people who have symptoms? Where does it occur in the gene? Certain areas of the gene are more accepting of changes than others. What do protein prediction models look like? We have computer programs that can help us interpret how the body might interpret the change. There is also likely pathogenic and pathogenic variants, which are where the lab has enough evidence to say, yes, there's a strong evidence that suggests that this contributes to symptoms in a person. So with the test results, we can have a positive result where we do have an answer and an explanation for somebody's symptoms. We could have a negative one where we don't have a genetic explanation based on the variants that were reported out, or we might have an uncertain one where we need to do a little bit more research to know for certain if a variant that was reported out is the explanation that we were looking for. Here's another way to look at it. So on the left, we have our negative results, we have our positive result, and then in the middle, sometimes we have a negative, sometimes we have a positive, sometimes we still need to do more work with our variant of uncertain significance. So a genetic test has varying things that are included, and almost always will you see these things on a genetic test. So the first would be the genetic test name. In my example, we have a comprehensive myopathy panel, the gene of interest, meaning that they found a change in it. So in our example, ABC1, the location in the gene. So I always think of C like coordinate. So on the 100th letter, where most people have an A, it was switched to a T, and then the corresponding amino acid change. So amino acids are the building blocks to proteins, and you can think of it like a beaded necklace. So each little amino acid is a bead that's stringed together to create a protein. And 3 of our letters go to combine together to make one little amino acid. So on the 66th bead or amino acid of our protein, where most people have an arginine, it was switched to a proline. And it doesn't matter the names of those. I know it's probably French to most people, but that's another way to identify a genetic change. Zygosity is referring to, of the 2 copies of the gene, is the spelling the same or different? So heterozygous means that they're different. So one copy of the gene has this spelling error, the other copy of the gene does not. And the classification of the variant that they're seeing, in our example, it's pathogenic. And then there'll also be a little bit more information, what does the gene associated with? So in this case, it's associated with the stellar personality. It'll also tell you the inheritance pattern, which in our example, autosomal dominant, and it will also give you a little bit of information on the variant itself. Has it been reported out? Maybe it's been seen in the general population. Maybe protein prediction model suggested is something that the body will not handle appropriately. So the big why why do we want to do genetic testing? Why should we push to have genetic testing performed? So genetic testing is really important. For one, it helps us better predict the course of symptoms. And from the clinician end, that's very helpful so that we know that we are giving the absolute best care that we can. We can predict maybe when symptoms might onset or when things might change a little bit more than you might have anticipated. In addition, it's also helpful for families to know what to expect. Sometimes just being able to prepare and knowing can make things a lot less scary and also make things a little bit more enjoyable. You're not going to be very concerned if this is normal or abnormal for you and your body. Something that is very important is eligibility for clinical trials. We are in the age of treatment. There are so many opportunities on the horizon for these amazing treatments, and almost all of them have to do with genetic backing. And therefore, you might be, based on a genetic change, eligible for a clinical trial. Maybe you won't be, but that would be ultimately for your safety. So knowing the exact genetic change and the exact gene that is involved, that is contributing to symptoms, can really benefit you and your treatment. Recurrence risk, knowing, what the chances are for future family members to have the same change can be helpful so that you can best prepare you and your family or this new baby about symptoms that they might occur over their life. Again, to make things a little bit more digestible and also to make things more normal. And the closure of a diagnostic odyssey, and this is something that should not be forgotten. Sometimes it can feel so overwhelming to go from doctor to doctor and not really have a why. And maybe you have a diagnosis, but you don't have a why. And closing that chapter can be psychologically beneficial, to say, like, I know what's going on. I don't need to know any more information about this. And I can, accept and continue on with that new information. And that, again, not to be something to, forget about. In congenital myopathies, there's something that we see called clinical and genetic heterogeneity. And that is also why it is very important to have a genetic explanation. As genetic testing has become more available, we can see that there are many genes that can cause many different clinical pictures. And with the clinical pictures that we're seeing, we can see that many genes can cause that clinical picture. So if you look at this little diagram, on the left is the structural differences that you'll see on muscle biopsy. In the middle column, you'll see genes that are known to cause those changes. And on the right column, you'll see the diagnosis name. So as you can see, there's a couple repeat offender genes on this list that can cause many different clinical pictures. And therefore, it's important to know, and you definitely cannot assume one gene over another based just solely on the clinical diagnosis or based on the structural difference you're seeing on muscle biopsy. And more specifically with Nemelin myopathy sometimes is a diagnosis that's given without any genetic testing being done because a muscle biopsy can show you Nemelin rods. Historically speaking, muscle biopsy was used to diagnose muscle differences. And what they would see, as you can see in the images on the left-hand side, are these little Nemelin rods. And as with the integration of genetics in our modern medicine systems, and the more we have used them, we have been able to better understand congenital myopathies as a whole. And we are also able to understand Nemelin myopathy significantly better with this genetic testing because of this heterogeneity, meaning that there is diversity amongst the group that we're seeing, or different categorizations. So here you can see that I won't give you all alphabet soup, but here are the genes that we definitively know are associated with Nemelin myopathy. And below it, you can also see the clinical spectrum. So even within the same family, of who have all the same genetic mutation, they can present a little bit differently from one another. And between families, you can see people who have the same spelling error looking a bit different from one another too. So there is a very large spectrum of genes that can be associated, and even within those genes or even those specific spelling errors, you can see a broad spectrum. Some other genetic phenomena that I wanted to review that you might see or encounter on genetic testing reports or when speaking with your doctors. Mosaicism is something that can occur. And if you think about a mosaic painting where multiple different colors come together to create one unified image, this is similar with our genetics. We can have more than one cell line that differs in genetic material that come together to make you who you are. So germline mosaicism is where you see different cell lines that have different genetic material in the egg or sperm cells. Therefore, if somebody were to get saliva testing for genetics or blood testing for genetics, it might not come back as them having a genetic change, and that's because it is within their eggs or sperm cells. Somatic mosaicism is where there is more than one cell line throughout the entire body. So this would be when a person gets a blood or saliva test for genetics, and it comes back that some of their cells have a genetic mutation and some of their cells do not. This can influence the way a person experiences symptoms based on the percentage of cells that do not have the mutation versus do have the mutation. And you may encounter the word confined, and confined means that there is more than one cell line in a single organ system. For example, the heart, or maybe the kidneys, maybe the ovaries. So that is, again, another term that is often associated with mosaicism. And then another word that you might see is variable expressivity, and that's just saying that symptoms vary greatly amongst individuals, and it could be amongst individuals that have the same genetic disorder, or even more specifically have the same genetic mutation. And that is all the information I have crammed together in a short amount of time. I know that genetics is really complex. Please do not hesitate to reach out to me for any further questions or clarifications.
Brianna, thank you so much. Really appreciate that. If you don't mind hanging on for maybe the Q&A toward the end, that would be amazing. I didn't see anything pop up in the chat just yet. So hopefully we'll get Carson rolling here with his own slides, and then we'll be able to loop back to the Q&A. Thank you.
Okay, great. So we're probably going to run a little bit beyond the hour. I hope that's okay. So thank you, Brianna, for this excellent introduction, because we're going to build on that in this talk. So I'm a child neurologist at the NIH and neuromuscular specialist, and also see patients with Brianna at the Children's Hospital of Philadelphia. So what I'm going to try to do today, and it's going to be a little bit of a tour de force, is tell you a little bit about what's there in the toolbox that we have at the moment. Let me see whether you can see my mouse, right? Okay. And how that would apply to the mammalian myopathies. Okay. So what do I mean by genetic toolbox? The genetic toolbox is really a toolbox of therapeutic approaches that are meant to go directly to where the mutation and the genetic origin of the disease is. And that is based on a paradigm and a promise, if you will, which is that the closer you get to the origin of the disease, the higher we expect the impact to be. And here is, again, a simple diagram that is based on what Brianna told you about the flow of genetic information, where we have the origin of our genetic information is encoded in the chromosomes, in the DNA, in the genes. And then there is transcription, a readout from the chromosomes into an RNA, which is the transcript. That's all happens in the cell nucleus. And then that is made into a protein. And the proteins together then make a person. Now, in a genetic mutation, we have an encoding problem in the DNA itself, and that's carried forward to the RNA that causes a faulty protein and in the end, the disease. And the promise of genetic therapies is really to move as close as we can to the genetic origin of the disease, that is the mutation itself, corrected up there to then cause an influence of the disease that makes the disease better. If not a cure, then at least a significant change of the disease. What does precision medicine mean in this context? Precision medicine means a treatment that's designed to specifically and precisely address the cause of the disease. So in this case, a genetic condition. So that's why it's so important what Brianna told you about in the first half of the seminar, which is we really need to know the genetic cause of the condition, not only the gene, but down to even the mutation level, the genetic change that's causing the disease. So precision diagnostics, if you will, is a precondition for precision therapeutics. The Nemaline myopathies you've encountered in the seminar series perhaps before or through talks by Alan Becks and his team are disorders of skeletal muscle, of course, and they're all based in a particular part of the muscle fiber, which is the sarcomere, the apparatus that is functioning to generate strength and contraction of the muscle. Not only that, they're in a particular subgroup of the sarcomere called the thin filament, and all of the genetic causations of Nemaline myopathy affect genes of the thin filament or of the maintenance of the thin filament, leading to the Nemaline rods. But this is really the only slide I have about this, because for thinking about genetic therapies, we don't actually need to think about the individual genes and parts of the body where that is happening. We are thinking about genetic mechanisms that are causing the disease. So that means going back to what Brianna said, what type of genetic mechanism is underlying this disease, and can we apply the tools across different genetic disorders to these particular causations of the disease? That actually is a strength of genetic therapies, which means we can apply tools across completely different disorders just based on their genetic mechanism, the type of mutation, and what is happening at the genetic level. And for the Nemaline myopathy, like for many other conditions, we can break it down in two basic, very basic genetic mechanisms that cause the disease. One is loss of function mutations on both gene copies, that's recessive disease. And the other one is dominant negative mutations on just one gene copy, that's dominant disease. And these are often causing a toxic interference and a disturbance of the cell by virtue of just being there. And if you then take the genes that are this alphabet soup of genes that are causing Nemaline myopathy, and thanks to Alan Becks and Vandana Gupta for this table, you can see there's many, many different ones that are all in this thin filament that I described to you before. It's maintenance, but they can also be broken down into their genetic mechanisms, in their inheritance. And you can see some of them are recessive and some of them are dominant. You can also see their size and frequencies here. And these two genetic mechanisms is really what's going to guide our genetic toolbox when we think about gene therapy considerations for these. So when we have two genetic mechanisms here, and this is of course a bit simplistic, but essentially two, then we have two basic approaches to these two genetic mechanisms. One is the loss of function, the recessive mechanism, when both gene copies are not acting properly, they're inactive, they're missing to the cell. And so here our approach would be to replace it with a fully functionally gene copy to make up or for the loss of this gene function, for the loss of the protein. On the other hand, in the dominant situation where we have a toxic interference, a negative function of the dominant protein that's encoded by only one copy of the two genes, we want to interfere with that. We want to get rid of the mutant copy and leave the functional copy undisturbed to do its normal function. So one is replacing, and the other one is inactivation of the dominant interfering form. So let's go for loss of function first. This is really the approach to many of the recessive loss of function disorders that we encounter across different genetic disorders. And here our recessive genes are highlighted in green, where we can think about an approach like this as being applicable. In my little diagram, I will show you, this is where AAV-mediated gene therapy comes into play. So here we use a virus, AAV, to deliver a healthy copy of that gene to the cell nucleus. And then it lives in the nucleus. It doesn't go into the chromosomes. It has its own transcript being made of it, and then provides a normal version of the protein that then is ameliorating the disease. So that's a concept of AAV-mediated gene replacement therapy, is to use an AAV, to use a viral envelope to deliver something that's missing or is not functioning properly to the cell. AAV is a virus. It's called adeno-associated virus, which is a virus that is not pathogenic for humans, but it does affect humans, infect humans. Just you won't notice that you've been infected with it. And it's called adeno-associated virus because it's very, very small and needs a bigger virus, a bigger partner, the adenovirus, to be fully functional. And it has a very simple genome here. Those are the genes of adeno-associated virus that are living within this little capsid, in this little envelope. And it needs more genes to make itself, and that's why it needs the helper genes from the adenovirus that comes along with it. And the principle of gene therapy is to replace these genes that are AAV genes with the gene that you want to deliver to the cell. And you just leave the envelope, you stuff it with the gene that you want to deliver, and then you infect the body with the envelope, and it can't replicate because it has no viral genes on board, but it delivers the missing gene to the cell where it's needed. So that's the principle of gene therapy. And you make this construct, the viral capsid, the envelope, and the transgene you want to deliver in the dish, basically in a bioreactor. It's an artificially made now hybrid virus, with a virus looking like a virus to the body from the outside, but delivering something that's missing to the cell on the inside. That's all good and nice, but the disadvantage of this is that what you can package in there can't exceed what the virus normally has on board. And so there's a size limitation to what you can deliver. And that's going to be important when you think for which gene this is applicable. And this is how you do that in a bioreactor. Basically, you have now the viral genes taken out, you put the gene that you want to deliver and that's missing in there, but then you have to supply everything else that's needed to make a viral particle in the system separately, and you put that all together. And then when the bioreactor puts this all together, you end up with a viral capsid made by the genes from the normally virus that you supply here outside. And then the transgene is packaged into that. So your therapeutic gene is packaged into that, and there are no longer any viral genes in this preparation. So that's your gene therapy product. And these capsids come in different versions, different serotypes that are often naturally occurring variations of the capsid. And those determine which cells are best infected by this envelope. These serotypes are currently naturally occurring serotypes, but they can also be engineered to be really good at infecting muscle, for instance. And so now we have serotypes out there, meaning capsids out there, envelopes out there that are particularly good at infecting muscle. And that is going to be a big step forward in gene therapy for Nemaline myopathy. So because this is a very generic packaging system, you can package anything you want into this viral capsid as long as it fits within the size limitations of the virus. And what I'm going to talk about next is really to deliver a missing protein to the cell. That is this transgene here, that's a missing gene, the defective gene that you want to deliver, and it's driven by a machinery that allows it to be made efficiently in the cell. So that's best for recessive loss of function disorders, as I told you, and best for moderate size of the gene message, so it can't be too big. This is another cartoon how this works. So here again, you have the envelope of the virus. This looks like a virus to the body. Body doesn't know what's in there when you infect the patient with it. And so the virus does its job. It goes to the cell, it infects the cell and delivers to the cell nucleus the gene that we want to deliver therapeutically. And it can't replicate because it has no genes of its own on board. So this is a one-time affair. It's infecting the cell, delivering the gene therapy product, and then you have essentially the machinery in place to make the gene that is missing. But on the outside, of course, as I told you, to the body, it looks like a virus. And so once you've given it, the body thinks, well, this is a virus, I better make an immune response against it. And so next time you would encounter the same virus, it would be neutralized by the body. So it's a one-time affair. Once you've done that, the body is immunized against the viral capsid and you can't do it again. So you have a one shot at it and you have to be really good at doing it. So amongst the genes where this would apply to the recessive genes, many would fit, but unfortunately one that is the one that is most common in the recessive forms of Nemaline myopathy, the nebulin itself, it is too large. So here, the genes that are listed at small and medium would fit into that, but nebulin, the big player by frequency for recessive Nemaline myopathy, is too large. It doesn't fit in them. That is not the end of it, because you can think about delivery tools that have no size limitations, such as AAV, such as, for instance, lipid nanoparticles, where you can deliver much larger messages to the cell. And that's a delivery tool that's, for instance, used in the COVID vaccination. So that's something that we think is coming to the clinic as well for gene therapy, where you can deliver much larger payloads, including payloads such as such as large as the nebulin RNA. So let's move to the second genetic mechanism, which are dominant negative mutations, where you have to get rid of the offender. You have to get rid of the mutant copy of the gene to let the one that is not affected do its job undisturbed. And often having half of what's normally in the cell is enough to make a cell happy and function normally. So therefore, this inactivation of the dominant mutation is the way to go in these. And again, we want to be as close as we can to the origin of the genetic disease, which is the genetic mutation itself, or the transcript that carries the mutation before it's made into protein. And here we have a second tool set, which is no longer replacing the gene, but is actually trying to address the transcript itself that has this dominant interfering bad mutation on board before it reaches the protein state, before it's made into protein. And we call that transcript-directed approaches. So here we're not directing the approach against the gene, but we direct the approach against the transcript, the readout of the gene before it is being made to protein. And some genetic conditions can be best addressed with that, including the one that affect splicing, meaning the putting together the genetic information in the transcript, and also some of the genes that are just too big for gene therapy. And it also allows us to make single patient precision therapeutics, which is really the ultimate boutique approach. And here in my diagram, this is how this works. So this time we're not doing a gene therapy, you're making a custom mutation-specific ASO or oligonucleotide, a small nucleic acid that goes into the nucleus and now corrects the transcript rather than the gene itself, but then will then lead to making a normal protein and ameliorating disease. So a very good example of that is to try to inactivate the mutation specifically, knock it down without interfering with the wild type mutation. So that would be potentially applicable to all of these dominant mutations that I'm showing here in the diagram. And I will show you an example how that's done for a different congenital muscle disease, congenital muscular dystrophy. Just give you an example how that could look like. We have molecular tools that are available that are capable of recognizing specific changes in the DNA, in the RNA, specific changes that are single mutations that are recognized by our ASOs, by our oligonucleotides, by our genetic probes. And then by marking the mutation in the transcript, they're capable of allowing the cell to inactivate the transcript. Here's an example of that from the congenital muscular dystrophies. Here we have a mutant RNA where one exon is missing, and we're designing a probe, an siRNA, a genetic probe, directly for that mutation that only recognizes the mutation, but not the normal transcript. And that allows us to take the transcript down, specifically just the mutant one that you can see down here as a smaller one. And as a result of that, we're allowing the normal copy to work uninterfered, to work unimpeded. And that allows us to have the genetic product, the protein, to reappear. So this is a cell culture where you can see here the green, which is the normal product, is missing. And now once I treat and inactivate the mutant copy and let the normal copy work unimpeded, the normal product reappears and can do its job. This is, of course, only working if the mutation can be directly addressed in a way that doesn't interfere with the normal copy of the gene. And that sometimes is really difficult. And sometimes you have to make it for just one mutation for one patient, which may be not feasible in the development of drugs. And so we can also think about combining the approach that I showed you first, which is the gene replacement, with a knockdown approach. And so now we are knocking down the mutant, the gene copy in the patient completely, the mutant and the wild type. We just take it out, we inactivate the gene completely, which is easier to do than being mutation specific. But at the same time, we're replacing it with gene replacement, gene therapy that I showed you in the first part. So now we have a combination of knockdown, but we don't have to be mutation specific. We just knock it down and replace it with a good copy of the gene. And so that is a way to try to approach this dominant negative problem without having to be mutation specific, which is just very tricky to do. And that would apply for a number of the dominant ones, including one of the most common, which is ACTA1. A second approach to go after transcripts is splice modulation. As you've heard from Brianna, splicing is a very crucial process that happens once a gene is read into RNA. The RNA has to be cut down to its functional appearance. Everything that's in between the protein encoding parts has to be cut out so that the message can be made into protein. That's called splicing. And there are many mutations that we now know of that mess up splicing and cause disease, and that are potentially addressable with splice modulation using molecular tools. And that is particularly true for the large genes with deep, with a lot of introns and exons that include nebulin, for instance, or titin is another big one where that occurs. So here the principle is to correct an abnormal splicing with a molecular probe. Again, I give you an example from the world of congenital muscular dystrophies. So here we have two exons and there is an intron in between. That needs to be cut out. The two exons have to be spliced together to make a normal messenger RNA. And in this particular case, we have a mutation deep in the intron that messes this up. There's now a part of the intron that is also spliced into the message, and that really causes disease. So we have to get rid of this part of the exon, of the splicing here, and make it back to normal splicing. And we can do that with the molecular probe that modulates the splicing. It goes to the part in the intron that is now faulty and that it is spliced into the message, and we can mask it to the splice machinery and cause this to be ignored, and so that the normal version of the splicing is made again. That's very powerful and is really only and totally dependent on the mutation and not so much on the gene itself, the type of mutation that causes this. This was the paper that we participated in, Alembex, as well as you can see here, that did that for a number of genes in muscle to see whether this type of mutation occurs in different muscle disease genes, and can it be really looked for, and can it be acted upon? And you can see here is an example where this happens in nebulin, and you can see an abnormal splicing in the nebulin gene with exon skipping. And we also have pseudo exon mutations in nebulin as well that can be addressed this way, as well as in other genes that are shown here. This is another nebulin case where splicing is abnormal in the intron, and you can redirect it to normal splicing using ASOs. So very powerful. If you find these mutations, and these mutations are difficult to find, and sometimes it needs additional genetic diagnostic tools that Brianna described, including RNA sequencing, which elevates this onto a research level. But it's moving into diagnostic testing in diagnostic labs as well. So really having a precision diagnosis is absolutely essential to designing a precision treatment in this situation. Let's move to some other genetic tools that are really tools more for the future, but that are moving into clinical consideration relatively rapidly. One is something that you've all heard about probably, which is the genome editing, the CRISPR-Cas gene scissor system. So what is this? This is a very clever system that is really was really invented by bacteria to defend themselves from their own pathogens, which are bacteriophages, to find mutations and find genetic differences and cut and inactivate them. What this really is, you can best think about it as a search function in a word processor. So when you click the search function in a word processor, you put a little string of letters in there or word that you want to look for in a big document, then you hit enter, and then the search function in the word processor will jump right to where the corresponding part is in your big text. So you put in a look for this particular word, and then even in a book, in a really complex text, you will find this very rapidly with the search function. This is what CRISPR-Cas does as well. You put in a guide. This is the search term that you want to use. And then in the genome, this guide will find the spot that corresponds to it, and then direct a gene scissor system directly to that spot. So as if you put your search function in, and then you hit the delete or edit function on your word processor to correct or delete that word. And that's what CRISPR-Cas does. It searches for a particular part in the genome that you're interested in this case, a mutation. It directs the gene scissor system directly to that. The gene scissor system will cut it out, and now you can either have it completely cut out and re-anneal it like taking out a word in a sentence, and have the sentence exist without the word that you deleted, which sometimes is helpful. Or you can even repair it, meaning you can replace it with a corrected version of the word, which is more difficult to do, but certainly possible. So in both cases, you have either a cut where the thing is annealed back together, or you replace it with the corrected version of the chromosome that you had cut. So this can be done again by delivering the gene scissor tools with the AAV system to the cell. You now use the AAV capsid again as your delivery tool, as your envelope. You deliver the CRISPR-Cas search and delete function, or search and replace function to the cell where it's looking in the chromosome where to cut, and then cuts the mutation, or it replaces it. And then you have the effective therapeutic effect. Just cutting is easier than replacing. And so, for instance, in the part that I showed you before, where we have something in the intron that is causing trouble, that we can actually cut out without causing any problems with the protein encoding parts of the gene, and inactivate it that way. So here, the gene scissor would be just used to cut out the mutation that's offending the transcript in the part of the gene that's non-coding. You just cut it out, and you're back to a normal transcript. Or you can replace it with a donor DNA, so the replaced part, and you cut it out, and you can't live without it, so you have to replace it with a normal version of the gene that can also be done in a repair process that's just not as effective and a bit more difficult to do, and that's why it has not been developed into clinical applications in in the body yet. So genome editing can apply to many types of genes and many types of mutations. It has a lot of application when the technologies expand at a rapid pace. So one has to still, of course, do a lot of homework in trying to establish its safety, because you only want to do the cut where you need it and not outside. In particular, if you want to treat an entire patient, not just a cell outside of the body, but if you want to treat the entire patients safely, you have to be absolutely sure that you cut only where needed and you repair only where needed. And also you want to be sure that you are doing it efficiently enough that it actually modifies the disease. There is a number of special developments of this system that are really developing at a very crisp pace, that are also dependent and taking advantage of the same system of trying to find something in the genome with that search function. So again, we're putting something in the word processor. We're looking for the mutation with our search function, but now we deliver different repair tools, not just the crude gene scissor that I showed you before. In this case, we are delivering actually something that is more elegant, that can repair just a single base pair mutation. So that's called base editing. A version of that that's even more complicated is called prime editing. Again, you're directing your search function to the mutation that you're looking to correct, and then you deliver an even more complex and refined machinery to repair it. All of that is moving for different genetic diseases, but because it's a universally applicable tool, tools like this may apply to even specialized conditions, rare conditions, such as the Nemelin myopathy. So we have to kind of keep our eyes open for these. Any development in them can be piggybacked, can be applied to our conditions as well. I will skip this for the interest of time, and just make one last comment here, which is that we can also use the CRISPR-Cas system, the search function, to modulate and regulate other genes that we may want to regulate in compensation for the gene that is defective. And so what you do here is you use our search function, the CRISPR-Cas system, to find a gene that you want to modulate. But now what you deliver is actually a volume switch, if you will, and you can dial up the volume or you can dial down the volume, meaning the expression of a gene, to make sure that you try to compensate for the gene that's missing. For instance, in ACTA1, you can try to turn the volume up on a different form of actin, the heart actin in skeletal muscle, to try to make up for the defective muscle-specific actin, as one example of that. So another clever tool where you direct this to a different gene that you then upregulate to compensate for the gene that's actually primarily defective. I mentioned skeletal actin and cardiac actin as a pair of genes that can be regulated in this way, tuning up the cardiac actin as a compensation. And in congenital muscular dystrophy, we have a similar situation. We have a gene that's defective, the LAMA2. We have a gene that is could compensate, but is not tuned up enough. And so here in this paper, this CRISPR-Cas upregulation system was used to upregulate the gene that could compensate, but is not tuned up enough, tune it up to compensate for the deficiency of the LAMA2 gene. So I end here by just showing you again, there is an ever-increasing, ever-complex therapy toolbox that applies to specific types of genetic mutations and genetic mechanisms that really apply across different genetic disorders. And therefore, that developing toolbox developed in all of genetic medicine can apply to the Nemelin myopathy specifically as well. So that is the power of this. We don't have to invent everything just for the Nemelin myopathies. We can really develop it across genetic disorders. And the future hopefully looks like you have a patient with a rare Nemelin myopathy, you identify the precise mutation, and then you pick the tools that best fit for that to treat the patient with that. And this only works, of course, as a team work, and hopefully we'll have ever-growing networks of special partners, special components that can provide the tools that are being developed across genetic medicine for our specific problems, in this case, Namelin myopathy. I do hope that this will be a powerful approach to these disorders in the future. And I will stop here.
Carson, thank you so much for that detailed and obviously brilliant talk. I would love to take some time to open it up to any questions that people may have. I think, there we go. Okay, thank you. Obviously, genetic variation is part of what makes each of us unique and even special. So I just want to make sure we acknowledge the fact that sometimes these variations are, again, what make us our own individual selves. And we want to make sure that we're placing these therapeutic targets in a position where we're trying to follow science, and that when individuals are ready to pursue therapeutics and the time is right for them because they're trying to seek out some opportunity for improved physical function, then we hopefully will be there. And Carson, it sounds like, Namelin myopathy is not the only one that is seeking this therapeutic approach, and a lot of science is in a race to solve the disease in a dish, sort of solving all these issues. And I think our goal as a foundation has been to keep researchers funded well enough so that they were ready as science presented these new therapeutic targets and approaches, so that, well, we would already know what has not worked and we'd be able to jump into new therapies. So in general terms, I'd love to see, yeah, go ahead.
Yeah, I think that to address some of the questions in the chat, the general point that I was trying to make, and I hope I made, is that the good news of all of this is that the development of genetic medicine happens across all of genetic disease. It doesn't have to happen anew in every single subgroup of genetic disease. So that's really the powerful message here. So we can use tools that are developed for something that's more common or that has more kind of financial power behind it. We can use these tools and then apply them to the much more rare and perhaps less well-funded conditions such as the Namelin myopathies. And really, that is a message that is hopeful, because it means that even a rare disease, even a single patient may in the end benefit from the development of a toolbox of genetic therapeutics that is developing across all of genetic medicine, if we put all the things in the right place. So gene replacement therapy is a good example for that. So what we've learned in other muscle diseases is certainly informing what we do in a much more rare disease, such as Namelin myopathy. And the same is true for the ASOs, for the precision therapeutics that we are sometimes designing against an individual mutation in even one patient. This is now no longer kind of futuristic. It becomes closer and closer to a possibility. And hence it's really important for every, even if it's not happening now, but it's important for every single patient with Namelin myopathy to try to work with the genetic caregivers, the genetic providers, to really drill down to the exact mutation that's causing the disease, if possible. It's not always possible on the first go, but the point is, if it's not possible, to not give up and then wait for the next genetic tool to come down the line and really revisit it. So it's an ongoing process. If your diagnosis can't be clarified in the first go with genetic testing, that's not the end of it. There is a constant flow of new genetic tests in research labs such as Banks labs that can pitch in and try to do this. Really, precision diagnostic is the first step to precision therapeutics. So we're in a place now to get everyone diagnosed, hopefully. Almost everyone diagnosed, hopefully, and that'll be the first.
We're going to try and review that here at the end. We're going to try and give people the steps that, if they're not genetically confirmed, that they can go through. So if you can stay on for that. But addressing Indira's question directly about NEB variant, will it take longer to develop since it's so large?
Yeah, the honest answer is probably yes, because it is it doesn't fit into the kind of simplistic gene delivery concept that I showed you before with the gene delivery with AAV just because of its size. And so nebulin is a more complex issue because it's so big and it can't be properly packaged into it. And it's also mostly a loss of function, meaning we need to supply better functioning nebulin to it. So there are, that's for nebulin as a whole, but there are certain specific mutations within nebulin that may be addressed earlier. There's a common mutation, for instance, exon 55 that's being addressed now specifically. There may be in a single patient a mutation that can be addressed very effectively by splice modulation, such as the pseudoexon mutation that I showed you, where you can design a precision splice modulator for that individual patient and treat that. For the delivery of the entire nebulin gene, we'll have to wait for better gene delivery tools, such as lipid nanoparticles, or for tools that allow us to correct genetic mutations in the DNA itself, the CRISPR-Cas1 that's safe enough to do. So yes, it's lagging a bit behind, but there's subgroups of patients that may benefit from these tools quicker.
And just to back up on and say again why the community can be helpful, if they get genetically confirmed, we'll know exactly where the gene location is that's affecting you. If you have that confirmation, also we could utilize any tissue sampling that would be offered to the tissue repository to offer to researchers to actually have the human cells in their laboratories to test with. We also have the CMDIR, the registry, identified with your confirmation. So we know if you check the box that says we can reach out to you, then we would love to be able to reach out to you. And tissue sampling, by the way, that we learned that it's not only important that you do it just one time, our bodies morph over time. And so having tissue samples, if you were to have an upcoming surgery, but you thought you already gave some tissue to the repository, we would please, Stacy Cossette would love to be able to help you with, she does a lot of the work for us and getting the tissue to the right place and stored appropriately. I think Aaron's asking an important question to understand, like, what's the actual procedure? I think a lot of us have received a flu shot, and a flu shot is carrying a very minimal virus as well, right? Aren't we getting some intervention that is somewhat similar? Carsten, can you explain a little bit about what it looks like?
I mean, in the end, the actual procedure that you have to undergo for this is relatively simple. It's an infusion in the vein, and so you get a lot of virus in there, and so the procedure itself is easy to do. It's an IV infusion, but then of course you have to make sure everything else that happens after that, once the virus is in the body and the body encounters the virus, is safe as well. And so at the moment for neuromuscular disease, we give really humongous doses of these viruses, but with the better capsid that are better at going to muscle, we'll be able to reduce that and do this more safely. But the procedure is kind of underwhelming, if you will. It's an IV where you get this medicine into the vein.
I'm just going down the list here. Are treatments ready for TN and T1?
Yeah, so that's one of the small ones with loss of function that actually fits into AAV. And so there is an active effort underway to make an AAV-mediated gene replacement therapy for TN and T1. That's led by Kevin Strauss, I believe. So they're working very diligently with academic and also industry partners to bring that to patients. So I hope to see that happening within the next 2 years. That's a great example where gene replacement therapy is applicable and where we can learn from any other gene therapy that's done in neuromuscular disease to really accelerate this forward.
And if I could just expand on that briefly, that was absolutely true. And it's being done, as you mentioned, by Kevin Strauss and a group at the UMass Worcester. There is another group of patients with mutations in TNNT1 where they're so-called autosomal dominant, passed down from parent to child. They survive longer. That's not applicable to this type of AAV-based gene replacement therapy. There is work in that area as well. But what Carsten described applies to the recessive form that was first described in Amish families.
Exactly. So that goes down to my breakdown into the recessive loss of function, where AAV replacement is applicable, versus the dominant form, where you have to get rid of the dominant offending mutation.
Great. The next question was from Carol. It was about the CMDIR, I believe. And yes, it is important to update occasionally, especially when you have a change in functional status. That is really helpful for us. We did move to a new platform that actually has a few more detailed and updated questions as well. So Sam Jacobson is actually making herself available to actually sit with people on the phone or Zoom for about an hour, walk people through the process of getting all of their information updated and as accurate as possible. Carson, I'm going to read this one out aloud. Laura Lou's asking a technical question. What is the infection efficacy of the current adenoviral system in skeletal muscle, and any strategy to assay off-target effect by gene editing?
Yeah, that's a good question. So it's adeno-associated virus. So it's a kind of small adenovirus was also used in gene therapy. That's a big guy that's sitting next to the small guy on my slide. So the big adenovirus was initially used in gene therapy, but had some real problems in the first phase of gene therapy. And adeno-associated virus is the one that we're currently using. So the infection efficacy for the skeletal muscle of the serotypes, meaning the versions of the virus that are currently used, which are naturally occurring versions of this virus, is okay, but it's not great. So you have to give big doses of this virus to get to the muscle effectively, and so you also overwhelm other organs with that, most notably the liver, and the liver doesn't need this and for the muscle diseases and often has a tough time dealing with it. That's part of the danger of gene therapy at the moment that we're trying to get a handle on. But there are now engineered versions of these capsids, of these envelopes that are made to go to muscle much more effectively. And so then the efficiency to go to muscle is an order of magnitude better than what we started out with, which means we can use lower doses, we stress the liver less, and we're more effective to go there. So that's a really big step forward. Hasn't been used in humans yet, but it's being developed by a number of different labs. There are strategies to assay for off-target effects of gene editing, and those are being refined and refined and refined. And so that's moving at a rapid clip. At the moment, gene editing is done mostly in cells. So you take, for instance, in a blood disease, you take a stem cell out of the body, you correct this stem cell, you very, very carefully check for off-target effect in a single cell. That's much easier to do than in a whole body. And then you put that corrected cell back into the body. It goes to the bone marrow and makes the good blood cells that are missing. That we can't do easily in muscle disease. So in muscle disease, we really have to go and edit all the different muscle cells separately. And there we are not as good yet. So in animals, the off-target screening is going on, and it still turns out that there is quite a bit of off-target cutting in the system. But the assays to do that and the refinement of being really just at the point of where you need it is getting better and better. So I suspect the better off-target screening and the better on-target activity will get us to systemic gene editing relatively soon.
Great, thank you. There's a question from Indira from South Africa, where I know one other child besides mine with NM. When treatment is available, will my child have access to it here?
All right, who wants to answer? It's a really good question, and I don't know the answer. Ideally, yes, I mean that's what I hope for the future, that there will be no geographic boundaries to it. At the moment, the reality is that probably these treatments are not yet globally available when they are developed. South Africa, of course, has a great center with Joe Windsors there, who is plugged in, of course, in all that happens. So I can see that happening there as well. For gene therapy, you know, one of the advantages, if you will, if gene therapy works well, it's not an ongoing therapy. It is a one-time therapy or few-time therapy that you do, and then you can leave the patient alone for, you know, years and years and years, ideally. And so meaning that a few centers could probably supply a lot of this to larger geographic areas. That's my hope in the future.
I just want to give a shout out to some of the researchers that we've already been funding in some of the CRISPR techniques. And so I don't know, Alan or Gus, if you want to talk through a little bit. We've funded over the years, the development of improved mouse models and all the different varieties. We've worked on zebrafish models and developing those as well. So I don't know if Gus or if Alan could chime in about who's working on these things and and future steps as you see them.
Sorry, Gus, I've been messaging offline with one of the participants. Do you want to start, and then I'd be happy to expand?
Okay, yes, we can say that we have been funding different projects internationally, and for example, we have a group now working with CRISPR technology in Australia. We have another group in Canada. As Mark mentioned at the beginning, Shane Dowling in Canada, he's working on one of the most common mutations in the nebulin gene with CRISPR. Of course, at the moment in human cells and animal models, and tweaking all these problems that we still find with these new technologies. And more classical gene therapy as gene replacement therapies. We have the group of Vandana Gupta and the group of Henk Rancière in Arizona, testing different gene therapies for some of the less frequent mutations like the KLH40 and 41. So yes, I mean, we have IFBS has been funding this preclinical developments since the beginning of the foundation, way before I started helping the foundation. And for example, another example that Dr. Bonneman made about using a different gene that you can upregulate to overcome some of the problems of the missing protein. This for the ACTA1 replaced by the cardiac ACTA1. The foundation was funding this work for a few years also, and still doing that. And the group in Australia that is now working with CRISPR, they are also trying to use CRISPR to upregulate that other gene, the cardiac gene. So yeah, I mean, we are trying to diversify our funds to look for all these different tools, how can be used, and which genes or genotypes can be used the best.
Yeah, so what I can just say on a very high level is that AFBS has supported efforts to identify therapies and be ready for trials on a very broad level. One of the previous webinars focused on clinical trial readiness and having information about Nemelin myopathy and knowing about enough individuals so that when a treatment becomes available, scientists, doctors, and researchers and companies can actually conduct the trial. And then, as Gus just described, the different approaches. So many of the different tools in the toolbox that Karsten was just talking about are being used by AFBS-funded researchers, such as CRISPR, such as AAV-based gene replacement therapy, such as potentially exon skipping. And then the last aspect is that they're not just focusing on one form of Nemelin myopathy. So the group in Australia is working on the actin gene and actin myopathy. Vandana Gupta, who's in Boston with me, has been working on KLHL40, one of the much more rare causes of Nemelin myopathy. There are researchers working on nebulin. And then finally, understanding and making progress on any one of these has the potential to help others. So a lot of the drugs or treatments that get developed are focused on one gene, or sometimes it's focused on one type of genetic variant or mutation, if you will. But in other cases, there are links and connections between these. So for example, KLHL40 is a protein that is thought to help regulate nebulin and help either stabilize it or make old tired copies of nebulin turn over so you can refresh it in muscle. And so to the extent that Vandana Gupta develops some therapy that might target KLHL40, there's the possibility, we don't know this yet, but it's a theory, that it might actually impact nebulin, which is one of the more common causes. So this is where I say that, you know, coming together under the AFBS umbrella, even though some people may have very sick, weak babies, others may be grandparents themselves who are still walking. Some may have one gene, some may have another gene. There's a lot of variation. Any progress we make in any area has the opportunity to help people in other areas. And that's one of the things I think it's important about AFBS and bringing everybody together.
Thank you. There's one question from Fahira that I want to make sure we address. Is there any clinical trials for ACTA1 ongoing? Any way we can participate? I already sent my child's report and muscle sample to CMDIR. Yeah, so muscle sample goes to CMDTR, just to be picky. CMDIR is a registry that has all your information where you probably sent your child's report, which are great. Two different things that we need to keep updating. So thank you for doing that. I know that there are currently no ACTA1 clinical trials going on, but there is a lot of investigation and a lot of discovery work, some of which both Gus and Alan just mentioned, and primarily out of the group from Australia.
Yeah, for all of the genes that Dr. Bonneman showed on that slide that listed all the Nemlin genes, there are not any specific directed trials, gene-related trials ongoing right now, but there are a number of different activities for many of these genes that we hope will come to trials sometime soon. So just stay engaged, keep asking those good questions. And I'll just say that, you know, there have been trials in related things. So spinal muscular atrophy, there's now an effective treatment. Duchenne muscular dystrophy, X-linked myotubular myopathy. Karsten's work is pioneering replacement therapy for something called giant axonal neuropathy. So there's lots of more examples. And as he pointed out, we're not working in a vacuum. So the Nemlin myopathies are one defined area that we all focus on and care a lot about. But we're learning from these other conditions and diseases, and advances there are helping us to make advances here.
Thanks for that. Indira wrote, and this maybe if one of the genetic counselors can help, Casey or Brianna. I have a 14-year-old who does not have NM. Obviously, her other child does. Is she a carrier of the mutation? If so, is there a way to remove the mutation so she doesn't pass it on?
I just told Indira that I gave her my email so she can follow up with me directly. But yes, lots of answers there, just specific to each type of gene and situation. So I can chat with her individually after.
And Brianna is also a great expert on the
Certainly.
on these earlier, Brianna.
I think, Stacy, did you want to make a quick comment?
Yeah, I just wanted to point out there was a comment from one of our participants that her daughter's going to be having a surgery. And it's really helpful if surgeries are not scheduled on a Friday. And that's just because we're at the end of the work week, and it can be very difficult for us to coordinate what needs to happen to get that sample preserved and shipped to us. We would not be able to ship that overnight on a Friday because no one is generally available at the lab on a Saturday to accept the shipment. So we'd have to have an arrangement made with the hospital where your surgery took place, where they're able to preserve it properly, keep it preserved at -80 Celsius until at least Monday. And not all hospitals have the capability to do that. So in general, I just try and encourage patient families that if you have a choice, try to schedule your surgeries earlier in the week so that we have a few days after your surgery to get all of this done.
Yeah, I totally endorse what Stacy just said. I'll just add though that if you do have to have surgery on a Friday, we'll make something work.
Out of respect for people's time, it's Saturday. Really want to thank you everybody for participating. We can end the recording. To the speakers, Carsten, Brianna,